What are you doing?
SATs.
Oh, okay. The SAT?
PSATs.
On Monday, what's for breakfast? What cereal? Is that Cocoa Puffs or something? No? Oh, Rice Krispies, chocolate Rice Krispies, what is it called? Anyway. So Monday, we have a quiz. Whatever we finish today is on Monday, okay? Two things on a quiz on Monday, it'll be 30 questions, joints and articulations, and everything about yesterday and today, okay? Behind me, we're talking about a neuromuscular junction, and if you look at this slide right here, there's a lot of terms that we talked about yesterday. So that included the ability of this nervous tissue, and there's a difference. Is there a question? When you're talking about nervous tissue, there's a difference between neurons and nerves. A part of a neuron is an axon or a terminal axon. So the neuron, terminal axon, and axon are all fall in the same category, and they carry electrical stimuli and they produce action potentials. So when you get to the level of the entire muscle, then you have nerves that serve the entire muscle, and nerves are something different. So don't use the word nerve, use the word neuron, axon, or terminal axon. When you get to the level of the terminal axon and that muscle gets really, really close to the muscle itself, there's a neuromuscular junction or a synapse. Those two things are very similar right there. A neuromuscular junction is an example of a synapse. There are many different types of synapses. The terminology that I didn't use yesterday, the part of this synapse that is before the synapse is called the presynaptic terminal, and then there's the postsynaptic portion of the meeting. The space is called the synaptic cleft, and if you look at the presynaptic terminal or the presynaptic axon terminal, that presynaptic axon terminal has synaptic vesicles. And if you look at my drawing yesterday, there are mitochondria in there, and then these little blank circles here. Those little blank circles were synaptic vesicles with neurotransmitter, one example of which is acetylcholine. Acetylcholine will come back to haunt us in Unit 4, and that's, we'll talk about it again. So neurotransmitters are going to be released through exocytosis under the stimulus of, and where is the stimulus here? From the electrical stimulus from these motor neurons. So the electrical stimulus causes the synaptic vesicles to be released, and that's called exocytosis. Through diffusion, it's going to move from an area of high concentration in the presynaptic terminal, cross this space, and it goes to this postsynaptic membrane, and that postsynaptic membrane is also called the motor end plate. That postsynaptic membrane is at the level of the muscle fiber, or the myofiber, or the myocyte. If you look at this term right here, and this is a term that I used in, I want to say in chapter 3, ligand-gated ion channels. Ligand-gated ion channels. An ion channel is a channel in a cell that accepts ions, particular ions, and a ligand-gated simply means that it's got special recognition abilities for a particular chemical. What chemical is it looking for? It's looking for acetylcholine. So here's an example of a neuromuscular junction. If you look in the upper right-hand corner, right here we have where my pointer is, we have those synaptic vesicles. There aren't any mitochondria drawn in here, but they're definitely there. When the electrical stimulus goes down the terminal axon, it causes exocytosis to crosses that synaptic cleft, and then endocytosis. So at that point, that's at the level of the neuromuscular junction. And now I talked about the sliding filament mechanism yesterday, or the sliding filament model. It's the same thing. We're now getting back to the muscle here, or the sarcomere. So sarcomeres shorten, and there's a couple different ways of describing how sarcomeres shorten. First of all, the thick myosin filaments, myofilaments, pull on actin. And I will say a bunch of different ridiculous ways of how they pull on actin, but they pull actin towards the M line, they pull actin away from the Z disc. That means the same thing. Okay? I am moving to your left, and I'm moving away from this wall. Both those things are correct, right? There are multiple ways of describing how actin moves in relation to myosin, how actin moves in relation to the rest of the sarcomere. And again, you have to be able to articulate that. As a result of the movement of actin, the sarcomere shortens, but the myofilaments do not. That's called the sliding filament theory or sliding filament model. And that's the basics there. When you have muscles that so now once the sarcomere shortens, how does it lengthen? Right? And the way that it lengthens is something has to pull on. So what pulls on it typically? Muscles that are opposite to one another. So where do you find muscles that are opposite? So a muscle in action, so what action is this? This is flexion and this is extension. So those are two examples of opposite actions or opposite movements. So the muscles that are strong flexors on the forearm, right there, include three muscles you need to know for Thursday and Friday. The muscles that are strong flexors are on the posterior portion of the humerus. Those muscles here, the biceps brachii, the brachialis or brachioradialis, those are antagonistic to the triceps brachii. So these muscles that have opposite action are muscles that produce the opposite effect, and what that does is it then allows the sarcomeres to lengthen, return back to original location, original form. Sarcomeres do not push, they only pull. If you look at this picture right here on the right-hand side, we have, and I have a better picture of this, but if I show you this picture up at the top, this is a photomicrograph of a sarcomere, an individual sarcomere. There's the Z disc, right down there, the M line. Here's the I band, right there, and then the A and H areas right there. If you look at this slide right here, it talked about a sarcomere shortening. There's multiple ways of saying this. So the H zone disappearing in a sarcomere, the only way that can happen is if the sarcomere shortens. The Z discs move closer together, right? The only way that happens is if the sarcomere shortens. Okay. So there's at least 4 different ways of talking about that. But visually, if you see this slide right here, let's just say I showed you this top part of the slide, and then this part, the bottom part of it, not the bottom part, but the middle part right here. I took out all of this drawing right here, all of this art. This is the Z disc, this is the Z disc, this is the M line, this is the M line. So in this case now, I tried to line them up. The M line is perfectly oriented there. And so where did that drawing that I drew yesterday come from? That came from this slide right here. By the way, we're up to slide 30 and we're still talking about that drawing yesterday. You should be able to identify everything in this top part right here, this top portion of the sarcomere here. You should be able to tell me how many sarcomeres are here. There's parts of 3 sarcomeres. There's one here. There's an entire complete sarcomere here. And then there's a 3rd sarcomere there. Down here, you should recognize that this is not a relaxed sarcomere. This is indeed a contracted sarcomere. And the adjacent sarcomeres are also contracted. How do I know that? Well, if you look at up top here, there's a lot of light area right here, there's a lot of light area right here, and there's much less light area down here. So what happens then is when your muscles contract, when a sarcomeres contract, that characteristic striations, that alternating dark and light area, that disappears or almost disappears. Again, another picture of it. The arrows there indicate the shortening of the sarcomere, and both Z discs move. When we talk about individual muscles, sometimes students get confused because I talk about the origin and insertion of muscles, the 2 connections to the skeletal system, and I tell you that one of them moves relative to the other. The insertion will move, the origin does not. In this case, both Z disks move. So now this gets a little bit into some neuron physiology, and so we'll talk a little bit about that, but I want to go very lightly on this, and so because we're going to revisit it in Unit 4. When you're talking about neuron physiology, especially motor neuron physiology, because motor neuron physiology is the only thing that we're worried about here. The motor neuron here has what's called a resting membrane potential, and the muscle cell does as well. The resting membrane potential is something that we have already talked about. The resting membrane potential is formed by active transport, and it is present because there's this sodium-potassium pump present. So particularly for neurons, the sodium-potassium pump is really critical. More potassium is on the inside, sodium is on the outside, and if and sodium's on the outside. And sodium then is found in the plasma. Potassium is found in the cytosol or the cytoplasm. If you do not have this sodium-potassium pump, this resting membrane potential set up, then you can't have an action potential, and if you can't have an action potential, you can't have a skeletal muscle contraction. Remember about the structure of these are just membranes, specialized membranes. These are just specialized cells. Right? Because they're specialized cells, we can go back to the phospholipid bilayer and the hydrophobic nature of it. We can go back and talk about transport proteins. We can talk about ligand-gated ion channels, and those transport proteins are another version of a ligand-gated ion channel in the muscle. And again, that ligand-gated ion channel is an integral protein found embedded in a phospholipid bilayer, and it is focused on an individual chemical. That individual chemical is a neurotransmitter. What neurotransmitter? ACH. Okay? So again, now I'm building on the vocabulary. This is from Unit 1. You still have to remember that stuff. The book says that there's two types. There's voltage gated and there's chemical gated or ligand gated. We're going to really focus on ligand gated ion channels. So for this portion, and I think in Unit 4 I have a little bit of commentary about voltage gated, but I don't worry about voltage gated ion channels much at all, except that they exist. For this quiz, this voltage gated portion of the slide will not be on it. So now we're going to get into what the cell looks like here. And again, now what the muscle, this is an individual muscle cell. If you look at the upper left-hand corner here, if you look at the lower right-hand corner, we have a combination of pumps and these channels. And once again, a channel is simply an integral protein. We're going to talk about how the muscle starts the contraction. When the muscle is starting the contraction, and if you look at this part right here, where this sensor is, right now, in this drawing right here, we have what's called what in our vocabulary on Thursday and Friday, this one will be called the dry earth strap. And now in this case, this drawing, you have an individual probe that penetrates an individual muscle cell. We're going to do that Thursday and Friday, except we're not going to penetrate the muscle cells. We're going to put those electrodes superficially, and it's going to detect the electrical nature of that. So when the muscle is not contracted, what is it going to say? It's going to say there's a resting membrane potential and nothing changes. This is all more description of that, right? The resting membrane potential and measuring the resting membrane potential. The resting membrane potential is negative, and the charge there is in millivolts. It's very, so 1,000th of a volt. When a neuron is resting, it is polarized. Right? I don't ask lots of questions about this, but when the neuron is resting and it's ready to send in its signal, it's polarized. If it's polarized, it's at this negative potential right here. And when it's polarized, it's ready to send a signal. When it's ready to send a signal, it's ready to cause the muscle to contract. When it sends a signal, that's called depolarized. So the only way the lights are on here is having electricity coming to the lights. When you turn those lights off, right here, then the lights go dark. So if you want to turn the lights on, you have to have electricity coming to the switch. Right? That's polarization. When you turn the lights on, you hit the switch, the lights come on, and that's depolarization. So depolarization is this, in this drawing right here, is this rapid increase right here, and that signal, what's going to happen at the level of the neuron? You're going to lose or release neurotransmitters from the neuron. What neurotransmitter? Again, acetylcholine. And then the neuron will return to its original resting memory potential. And I won't ask you about that, but depolarization is something. Polarized neurons send signals. Depolarized neurons are in the process of sending. Wait a minute. Polarized neurons are ready to send signals. Depolarized neurons are actually sending signals. And when they're sending signals, you're talking about it on the scale of a millionth of a second or a hundredth of a thousandth of a second, very, very rapidly. So you see this here, milliseconds here, 2 milliseconds, 3 milliseconds, really, really quickly. So when a neuron is ready, it's polarized. When it's actually sending the signal, it's depolarized, and then it returns to being polarized. So in the process of sending those signals here, what's happening? It's movement of sodium and potassium. How do you know where sodium is going to move and where potassium is going to move? Well, you simply set up the sodium-potassium pump originally. Sodium is outside of the cell, so where does sodium move? Well, it has to move inside the cell. Potassium is set up inside of the cell by the sodium-potassium pump, so where does it move? It has to move outside of the cell, and that's what this says. When it has to return to its resting membrane potential, and so then the sodium-potassium pump reconfigures it. It sends all the potassium inside the cell and all the sodium outside of the cell. Once again, we're not worried about the voltage-gated ion channel. The book talks a lot about this portion right here. And I think that this is too much on the nervous tissue, so I'm going to skip this stuff. The one thing I will say is that when you're talking about an action potential here, what we're talking about here is a series of changes. And if you look at this is the level of the axon terminal right here. And the axon terminal is sending signals from the center of the cell of the neuron down toward the muscle. And so in this drawing right here, this is where my pointer is far away from the muscle. And if you go down here in the lower right-hand corner, that's really much closer to the muscle. And that's really where we're interested in. So we're really interested in this diagram right here. So we were just worried about the neuromuscular junction, the action potential getting propagated down the axon terminal. And now we've already talked about some of this here, this structure right here. So one thing that is missing from this slide right here is the presence of mitochondria. The mitochondria have to be present throughout the axon terminal because the sodium-potassium pump has to get set up. In the left-hand side, you have an example of exocytosis right here. And now, if you look over on the right-hand side, and first of all, this is a neuromuscular junction. On the left-hand side of the diagram here, we have the verbiage here, the terminology associated with what I'm going to tell you about. First thing here is the ligand-gated ion channels, the signals are going to get sent down here. And through exocytosis here, we have the release of acetylcholine. That's going to cross that synaptic cleft here, and acetylcholine is going to bind to that ligand-gated ion channel. This is the channel, and it's specialized, it's focused on one chemical only, and that's acetylcholine. And then what happens? It opens up, right? And when it opens up here, then we start getting ready for a a muscle contraction. Now, look at this. I don't have to contract my muscles, right? I can control that, whether the muscle is contracted. And yet, there's acetylcholine floating around in my muscles. So I have a muscle contracting, and now I want it relaxed. Do I want the muscle contracted? Whenever acetylcholine is present in the muscles, right, then it's going to cause a contraction. So you want to turn that off. How do you turn that off? Well, there's a couple of mechanisms here. This thing right here is called might be in the next page, acetylcholine esterase. And you see this thing right here? Acetylcholine is released into the synaptic cleft. That's what I just said right here. But how do you stop acetylcholine from activating? So there's two mechanisms. One is endocytosis to the neuron. Right? So in other words, the neuron itself, the terminal axon, will suck up as much ACH as it can. And then the other thing is that acetylcholine esterase will grab ACH and it will break it down. So if it grabs it and breaks it down, it can no longer act, and then your muscles can relax. Once there's no more acetylcholine, or there's not enough to cause a reaction here or a muscle contraction, the muscle doesn't contract anymore. So there's two mechanisms to turn off the muscle contraction. The one is, now there's two different versions of endocytosis. One is endocytosis to the muscle cell, right? And that's down here in this neuromuscular junction. And the other is endocytosis of ACH back into the terminal axon so that it can be prepared to do another muscle contraction. Right? Now that's really critical because otherwise you can't turn off a muscle contraction. And if you can't turn them off muscle contractions, that's sort of a pain in the ass, literally. Right here. So on the left-hand side, we talk a little bit about the sodium here. I'm not as worried about sodium. That's that's at the level of the neuron. I am worried about this one right here. There is acetylcholine and acetylcholine esterase. This capital E after ACH means it's the enzyme, and that enzyme breaks it down into two parts. The two parts are acetic acid and choline. So now you break those two things off. You have a Lego, you break the Lego apart, now you have all the component parts. You can reform that Lego, right? And now the body does that in the span of milliseconds, okay? So then you're gonna have acetylcholine esterase that breaks it down, and then you can put it back into the neuron, another form of endocytosis, and then you encapsulate it in a synaptic vesicle and it's ready to go. So acetic acid and choline are recycled here by the body. Sometimes you can make it easily, other times you can't. So you want to be able to use that. Really quickly. So now we're getting down to the muscle cell. So that prior several slides were about the neuron, and that's something that we didn't talk about yesterday. But yesterday's slide gives you an idea of some of the things, some of the complexity, some of the details that I just talked about. Bless you. So that when you talk about the action potential itself, the action potential is what we were just talking about here. The action potential produces changes in the sarcolemma. And where's the sarcolemma? That's the cell membrane of the skeletal muscle cell. It then propagated or sent to the T-tubules and the triad, which is part of the sarcoplasmic reticulum. And that sarcoplasmic reticulum, sarcolemma, right, those are all sort of intertwined. The chemical that I'm particularly interested in here is that calcium cation, then it resides in the terminal cistern. And when it's released there, it releases, where does it get released? It releases into the sarcoplasm. And through diffusion, it binds to the G-actin protein, particularly the troponin, the G-active site rather, the active site. And troponin is where it's going to bind to. Troponin has three parts there. There's the tropomyosin is the fibrous part, the troponin tropomyosin complex there. and then that precipitates or causes a muscle contraction. And so here is some of the details of that. The one thing that I don't want to talk about all of this stuff. This is more for you to read if you want to get into more the details right here. So for instance, have I ever talked about the sarcolemma and the T-tubules? Have I ever talked about the SR and calcium ions? Have I ever talked about myofilaments and then cross bridges? Yes, I have. So I definitely talked about all of this stuff. A cross bridge forms, or cross bridges, multiple cross bridges form when the troponin tropomyosin complex moves off the active site of actin, and the myosin head then can grab or can grab the actin. When it grabs the actin, then a contraction happens. Now, at this part right here is, this picture is part of the, up close and personal with the individual sarcolemma right here. And once again, there's going to be a superficial endomysium right here. If you look at this top of this picture, this is called the cross-bridge movement, and this is at the level of the proteins causing movement at the sarcomere. Okay, so if I'm oriented back, that's the M line. I'm a myosin, and this is my myosin head. I'm ready to grab an actin. When I grab an actin, I pull it towards the wall. That's the M line, right? When I'm done pulling, I can't pull anymore. My ATP runs out. What do? I let go. If nothing else is attached to actin, then the troponin tropomyosin complex. Do you have a question? If that, yeah.
Oh, no.
If the myosin, once the myosin head is done pulling, it lets go. The troponin tropomyosin complex covers that G site again, and you need to get another one exposed. What else has to happen? You need more ATP. When there's more ATP around, then I get ready to grab. If there's an active site that is exposed, I can pull it again. More ATP, I can pull it again. I can pull it again. Right, so individual myosin heads will pull multiple times, but for each ATP, it only pulls once. This picture right here is showing, this is the myosin head grabbing the exposed G-actin site, forming the cross bridge, pulling it. That's called the power stroke. When it's pulling it, it then runs out of ATP and it releases. At this point, new ATP is formed. When new ATP is formed and present there, then the recovery stroke. So in this case, I grabbed the actin, I pulled, then I let go, the ATP drops off, I get, or the ADP and inorganic phosphate, sorry, a new ATP gets on there, and then I can get ready to pull again. It's only when the ATP is present and can I pull, and the active site is exposed. And this is what we're going to talk about right here. So I'm not going to get into the details of this anymore. I just told you about that. So we talked about troponin tropomyosin, how we talked about the active site in the presence of calcium, how we talked about ATP. This is the myosin head, and this is the myosin tail, right? So we've talked about all of this. Cross bridge formation, the power stroke. Now, if you notice here, this is, in the previous slide it said ATP, and this slide says ADP. Where did the other inorganic phosphate go? It went to the head of the myosin, and that head of the myosin now is pulling, and that's called the power stroke. When that power stroke is done, then the myosin head detaches from the G-actin site, and when the detachment then causes, or it allows for another ATP molecule to bond to it, and when it bonds to it, then it releases, it detaches, and it can get ready to grab again. And so that detachment then, with the return of the head, ready for another power stroke. So that's a single cross bridge movement. Right? That recovery stroke is the end of this movement. Right? Once the recovery stroke happens, then it's ready to pull again. So during a single muscle contraction, you can have the same myosin head pull multiple times, and there are many, many thousands of myosin heads. So it's not like we're talking about one myosin head pulling, 52 times. So this is a summary of a skeletal muscle contraction here. If you look at all the details right here on the, these are 9 steps right here. In the lower right-hand corner, I'm on slide 57 from today. If you look at all of what we talked about yesterday, we're still talking about what we're talking about today, right? There's some details that are definitely more specific. Right? What are some of the things that we talked about yesterday here? The influx of calcium from the result of the voltage-gated channels opening from the presence of ACH right here. The triad opening, the sarcoplasm accepting the calcium, right? ATP being broken down. Have we talked about that? Yes, we have. So so far, as near as I can tell, everything that we talked about yesterday is a repeat today. When you want to, once the muscle is contracted, you got to return to its original position. You have to relax that muscle. If you can't relax it, then you can't get ready to contract it again. The same thing is true with the neuron. If you can't return it to original position or original resting membrane potential, you can't send another signal. So the resting membrane potential in the myofiber also has a resting membrane potential. It has the sodium-potassium pump that is going to, again, it's sending out sodium into the cytosol, out of the cytosol, and it's importing potassium into the cytosol or the sarcoplasm or the sarcosol. ATP is required to detach the myosin head, so ATP is required for that. And you also have active transport when we release calcium from the terminal cistern. You got to get calcium back up into that terminal cistern before the muscle can recontract. Right? So that's another example. That's another thing that has to happen here. So we have sodium-potassium pump reconfiguring the sodium-potassium concentrations in both the neuron and the muscle fiber. And then there's also active transport sending calcium back up into the terminal cistern. If those things don't happen, you can't have another muscle contraction. And I think what we're going to do now is I'm going to talk for a little bit, and then we're going to stop here. And we're going to end in a couple of minutes. This part right here, or whole muscle physiology, have you ever seen EMG, electromyogram? Not yesterday. You saw it last week, okay, in lab. So what we're talking about in the next couple of slides is for this week in class, in lab. We're not talking about on the quiz, okay? I won't ask the whole muscle physiology and EMGs on the quiz, okay? So muscle fibers, we talked about contractions, or another way of saying that is the muscles can twitch. And that muscle twitch there at the level of the myofiber, that single twitch will be a result of the motor neuron. This is familiar stuff. There are multiple parts to this, and if you look at this graph right here, there's time in milliseconds, and we don't really have the milliseconds here, but we have tension. And tension means simply if my biceps brachii is to its longest extent here, I'm not pulling on anything. So at this point, my biceps brachii is completely relaxed, the tension's 0. Now I'm pulling my radial tuberosity up, and we're causing a contraction. The tension then is increasing. This myogram has 4 parts here, and only 3 are shown here. There's the first phase right here is the stimulus phase, where the stimulus is applied right there. Where is that stimulus coming from? It's coming from your motor neurons. So the stimulus applied, and at some point there's a short period of time when the muscle doesn't react. What's happening where my pointer is, all of the stuff that I talked about in a muscle contraction here, where you're sending the signal, the ACH is crossing that synaptic cleft, the calcium's getting released, the calcium's diffusing, all of that stuff is happening. Cross bridges are forming, ATP is there already, right? The G-active site is getting released, is getting uncovered here, the cross bridges, all of that stuff's happening during this point right here. That's called the lag phase. When the cross bridges are formed and then you start pulling here, that's the contraction phase. And then there's the relaxation phase. That's when the muscle is getting stretched by another muscle. And then it's returning, while it's doing that, it's returning through active transport. It's returning calcium back to the sarcoplasmic reticulum. Again, SR is sarcoplasmic reticulum. This contraction phase is what we've been talking about right here. So this EMG is what we're really interested in. If let's just say I had one laptop. I don't feel like picking up a laptop. If I had one laptop, and you're going to be asked to do this this week. So one laptop, and I'm going to ask you to contract your biceps muscle. You're going to measure the EMG and those biceps muscle, and then I'm going to say, do that with two laptops, and you're going to measure it, right? Now, if I were to say do one laptop and keep it there for 3 minutes, right? That might be a little hard. Your entire muscle is not contracting originally. Your entire muscle has these things called motor units. Right? Your entire muscle, this is under voluntary control. And you're saying, I want to keep this laptop like this for 3 minutes. So some part of the muscle is going to contract, and the other parts are going to relax. So this part right here, everything controlled by this one neuron is contracting. Everything that's not controlled over here, this is not controlled. That's not going to contract. So now there's multiple motor units in an individual muscle. So there's a motor unit that's contracting. The others are relaxing. What are those relaxing doing? They're getting ready for a muscle contraction. Now this one runs out of energy. So does the muscle fail? Well, hopefully not, because you have other motor units that are ready to fire. Those motor units then ready to fire, the first motor unit relaxes, and it gets repaired. It repairs itself. It returns everything to resting membrane potential, and it repairs itself, get ready to fire again. So now you have multiple motor neurons there that feed a single muscle cell, and it can then control those motor units. The bigger the muscle, the more motor units, the more control you have.
Sure, thank you.
Unless you're talking about small muscles with delicate muscles. When you're talking about the hand movements right here, they're not very strong muscles, but there's a lot of control there. The control there is because of the motor units. Let me just see something here. And, all right, so that's enough. We're going to stop right here. So just to review here, everything up to this slide right here in motor units will be eligible for the quiz on Monday. The quiz will be on 2 parts there. There's joints and articulations and muscle tissue. Once again, we're on slide 60, and now we're into new material right here. So I would say roughly around 40 slides that I got through yesterday in class from a single diagram. Once again, I'm going to update those diagrams that I drew yesterday. I'm going to post them online, and they're going to be in the Unit 3 diagram section. Are there any questions about what we're doing on Thursday? So literally, I'm going to have you guys ready. I'm going to have all the equipment ready for you to use. I'm not going to tell you what to do because you already know how to do it. And I'm going to just say, look at the diagram, read the directions, And by the end of lab on Thursday or the end of lab on Friday, I expect that you have the summary submitted. And before lab, or before midnight tomorrow, there's an individual muscle, there's a lesson due. I don't remember the name of it, but there's a muscle lesson due. Or before midnight tomorrow. Bless you.
Thank you.
Okay, are there any questions for me?